The prevalence of infectious disease worldwide has decreased dramatically during the last decades and continues to decline. However, the decrease of infectious diseases goes hand in hand with an increase in autoimmune diseases and allergies. This is called the hygiene hypothesis. These diseases are already a major problem in developed countries and are rapidly becoming more common in the urban areas of developing countries. When facing such an increase in welfare diseases, we need to ask ourselves, how can we produce enough affordable drugs to treat these diseases? How can we adapt to the growing market? What is the best production method for this medicine? These challenges need to be met, presenting an opportunity for visionary companies willing to embrace new methods to produce the solution for this problem.
Antibodies currently form a large group of biopharmaceutical drugs, and the production of new antibody-based pharmaceuticals must increase to meet future demands. Right now, Chinese hamster ovary cells, or CHO cells, are most commonly used for the production of these antibodies. An important advantage is that it is an established system, making it easier for new drugs to get FDA approval. This makes it the dominant system for pharmaceutical companies. One of the downsides of using CHO cells is that production takes long and is costly. A major drawback of mammalian cell cultures is that they are vulnerable to infection with mammalian viruses and pathogens. Furthermore, upscaling takes time and may lead to alterations in the final product that have to be corrected.
In recent years, plant-based systems have gained attention for pharmaceutical protein production using either whole plants or cell cultures. Whole plant systems are scalable and cheap. They utilize Nicotiana benthamiana, a wild Australian relative of tobacco. This system does not require genetically modified plants, but relies on transient expression. This means that plant tissues are infiltrated with the bacterium carrying the genes required for the production of pharmaceutical proteins. Within 5 days, massive amounts of protein can be produced with this system. Plant-based systems offer several advantages over mammalian expression systems. Firstly, plants are free from human pathogens and viruses. Secondly, plants require only water, light and nutrients to grow, which significantly reduces the cost of producing antibodies. It was estimated that a Nicotiana-based pharmaceutical facility would only cost half the initial startup investment and half the cost during production compared to a CHO-based facility. Finally, the major advantage of plants over CHO systems is in a significant reduction of time during the discovery phase of new drugs. Nicotiana systems utilize transient expression, which helps avoid the long development of stable transgenic cell lines.
Production of pharmaceutical proteins in plants has one major drawback. Most of these proteins, including antibodies, are glycoproteins. This means that sugar groups are added in the final step of protein synthesis in a process called glycosylation. The sugar groups added by plants differ from those added in mammals. These differences can lead to reduced functioning of the pharmaceutical protein and possibly even allergic reactions. This was initially the reason plant-based proteins were not approved by the FDA. The FDA wanted to first have more evidence about glycosylation. Researchers have since developed plants with adapted glycosylation pathways. This allows them to make fully human-like glycoproteins. The plants themselves do not seem to be affected by these changes.
In conclusion, these modified plants are capable of producing proteins with human-like glycosylation patterns. Recently, L-lyso, a plant-produced human glycoprotein, was approved by the FDA and is now widely used to treat Fabry disease. This marks the first step for a wider application of plant-based systems, which are shown to be cheaper and faster. The discovery phase for new drugs in particular is greatly reduced, ensuring faster production of novel drugs. Plant-based production systems can be used to supply an adequate quantity of medicine to a growing market and increasing number of patients.